Dear students, welcome to the second lecture on biosensors. Today in this particular lecture we are going to recover different kinds of biosensors and what differentiates them from each other, and how we have utilized a wide array of physical and chemical properties to be able to understand what kind of contaminants are present in our environment, and to get rapid information so that we can take quick actions, we can make quick, well directed and informed interventions to tackle environmental health and public health problems. So let us get started with biosensors. Alrighty, as a recap, remember what biosensor is. Here you have your analytes in the water, or here we have analyte in water or in sample. So these are your targets, and then here you have bioreceptors. So this is the shape of an enzyme, by the way. So you have bioreceptors, and it so happens that your target is a perfect match for your bioreceptor. When it matches, it undergoes conformational change in physical properties, chemical properties, or so on and so forth, and when it undergoes change, this change can be converted into an electronic signal. This entire business is part of job of transducer, and then the electronic signal can be output as either information on a screen, or as a light, like red light not good, green light good, something like that, or it can be read by a computer, which gives us meaningful data. Alrighty, now but where are sensors important? What is the scope for them in the market, and how would this help you in your career, in your entrepreneurship? Well, sensors have at least three very well recognized markets. One is the diagnostic market. This is a very well established, very large market in our country, and we have lot of foreign players entering our market right now, selling our public health bodies one of the most advanced biosensors and sensors we have for detecting public health, environmental health challenges. So there is lot of scope for developing new sensors for the diagnostic market. Now when we talk of diagnostic market, basically we are trying to get an idea whether the contaminant is present or not, whether target is not present or not. So this is diagnosis, and diagnosis is not limited to hospitals, and which is the second point actually. Clinical testing, which is diagnosis in context of hospitals or clinics. So definitely diagnostic market, for example, I want to know whether a particular milk has a particular protein or not. Does it have A1 protein or does it not have? I might be interested to know that after I have done a particular experiment on potato, is a particular protein still present or not. So all these kind of diagnoses can be done using your biosensors, and there is a big market for it. And when this kind of diagnosis is done in hospital clinical setting, you have clinical testing, and this is no doubt the biggest diagnostic markets. Then there are other markets, and nearly even the medical arena, we have lot of diagnostic market in agriculture, in veterinary, so and paramedical sciences, and no wonder definitely environmental sciences. Alrighty, so what are the requirements of sensor? What kind of sensors do we require? There should be relevance of output signal to measurement environment. So whatever I am measuring in the environment, whatever kind of environment I am in, the output that I get should be relevant to it. Next is accuracy and repeatability. It should not be so that once this sensor gives me a value of 2, next time it gives me 200 and then gives me 2 crore. I have no idea what to believe. So it should be accurate and it should be repeatable. And now accuracy, there is a difference between accuracy and repeatability. Perhaps what we are mentioning here is accuracy and precision. So not only should I get similar values every time I take measurement, but also it should be close to the actual value, the true value, which is accuracy. Next is sensitivity and resolution. So sensitivity is the ability to detect lower amounts of analyte. For example, if I have only 10 to the power 2 gene copies of vancomycin resistant gene per ml, which is very low concentration, if I am able to detect it, then I have high sensitivity. So sensitivity is what is the lowest concentration you can detect and get a good signal. Now resolution is, especially if you are how clear is your signal. As if we have multi-analyte system, if there is a biosensor that can detect glucose, it can detect sucrose, fructose and other kinds of sugar, pentose, then How well it can tell me? Well, the detection I am getting is of pentose, not of fructose. For example, if I have a sensor that measures that detects MRSA, it detects VAN-A like we talked about in the last class, and it also detects BLA1 gene, beta lactamase A1 gene. Now, so very good, I have a sensor that can detect all 3 really bad antimicrobial resistant genes in one go, but let us say it gets a signal. Yes, I have a signal. How well can it tell me, with what confidence and clarity can it tell me that this signal belongs to BLA1 and not VAN-A and MRSA? So, this clarity, it comes with resolution. So, we definitely want our biosensor to have very high sensitivity, which means very low detection and quantification levels, and very high resolution. Next is dynamic range, then we have speed of response. We have talked about this in previous lecture too, that we want our biosensors to give us as rapid data as possible, especially when we are suffering from public health epidemics and we have serious environmental challenges to take care of. Next is insensitivity to temperature or temperature compensation. Now, one thing is there in a country like India and many countries around the world, we have temperature variability in both diurnal cycle and also seasonal cycle. So, for example, where I am right now in North India, in winter the temperature might fall down, in Uttarakhand where I am right now, the temperature might fall down to below 5 degrees Celsius, and then during summer we might stay consistently 46 degrees Celsius plus, which is quite a big range of temperature. So, my sensor should not fail at these extreme ends. It should not go kaboom at 48 degree, 49 degrees Celsius, or it should not stop working at very low temperatures. Now, this brings me to a very important point. We are talking about biosensors, right? So, biosensors have biological agents that act as receptor. Now, for example, I have enzyme, but this enzyme is only very specific at particular temperature. You increase the temperature, it denatures, and you can't detect anymore. So, this is one limitation with certain kinds of biosensors. So, with biosensors, this is very, very important. It should not be sensitive to temperature. Even as temperature is changing, it should be very reliable. And even if the data moves with temperature, let us say it overestimates at higher temperature or underestimates at higher temperature, there should be a way for us to do temperature compensation. There should be a way for us to correct it mathematically, reliably. Which brings me to another point. I hope you can see this here. If you are talking about temperature changes, we are not expecting our biosensors to be sitting in controlled temperature rooms, laboratories. We are ideally looking for sensors that we can actually carry, take them to environment, get the data right there and right then. That is the demand of today. So, for that we really need that our sensors should be insensitive to temperature or should have ability for temperature compensation. Next is they should be insensitive to electrical and other environmental interference. For example, let us say in lab you had a sensor to detect MRSA, VAN-A and BLA1 gene. Very good. It worked perfectly. Now, you go to Upper Ganga Canal in Roorkee, or you go to river Yamuna, or you go to river Tapti is pretty clean, but you go to other rivers that are in really bad state, and then what you notice is that it's going haywire. Your sensor is giving you data that does not make sense. Why? Perhaps because it is quite possible actually. It's quite possible that you have other interfering agents that are not able to give you good data. It's also possible, for example, if we have a magnetic based or pH based or conductivity based assay, conductivity based transducers, then we have electrical interference and then things might go haywire and we might not get reliable data. So, regardless of what is happening in the environment, regardless of the temperature, your biosensor should be able to give you a very reliable information. Now, let us look at the 8th one, amenable to testing and calibration. It should be very easy for us to test whether it is giving the right data or not. It should be easy for us to test the value of inner sample. We should also have the ease of calibrating it. For example, most of the students here in this class would have at some point in their life done pH analyses, which is basically dipping the pH electrode and finding out the pH of the liquid. Now, if you remember, ideally before you dipped in your pH electrode into the sample, you calibrated it using pH 4, and depending on what kind of analysis you are doing, a higher pH solution, which you know is at pH 4 or at a higher value of pH. So, once you have two calibration points, you can be more sure about the data that you are going to get. So, it should be amenable to calibration. Next is reliability and self-checking capability. Again, same thing over and over again. I should be able to trust the data because it is repeatable, it is accurate, and it has shown its mettle over and over again. It should have a self-checking capability. It should be able to run auto check and find out what the problem is, if it's working fine or not. So if there's a problem but I don't detect, let's say I'm in the field and I'm measuring pH is equal to 9 of water and I'm surprised, but I'm like, well, the instrument is very reliable, and I record it, and then we inform the civilians and the government agencies, and people get concerned. However, if the sensor has a self-checking capability, it can say, wait, something is wrong with me, take me to the mechanic first. So that kind of information is helpful. Next is physical robustness. Again, as I was mentioning earlier, that our target perhaps now is not to have instruments, sophisticated giant sensors that sit in the labs, national laboratories, regional laboratories, but we want sensors that we can actually carry in the field, carry in the villages and get the data. For that, it is very important that the sensor should have physical robustness. Let's say I need to ship the sample, the sensor from Chennai to Delhi. Now, in order to do that, it will go via air mail or via train mail, however it go by road, however it is transported from Chennai to Delhi. If it does not have physical robustness, it's going to arrive damaged, and that's going to be a loss to economy and also loss to environment and public health, because we cannot detect contaminants that we could have detected by using it, and thus physical robustness is very important. Also, for example, I am working with Namami Gange and I take my sensor and I take it to my village, which is some 30 kilometers from here, and by the time I reach there, because the village roads are not very good, my sensor is damaged. That we do not want. Next is service requirements. Its service requirements should be minimal and the service should be very simple, because we notice that in many parts of our nation, many labs, many regions, many research facilities, servicing annual maintenance and is very challenging. Next, capital cost. Obviously, we don't want it to be very expensive, and running cost in life, we want it to have higher life and low running cost, and the acceptability by user. It should be something that people want to use. And then product safety. Sample host system must not be contaminated by sensor. So now this is very important. So the safety of the sample, safety of the host is very important. Sensor should not endanger the health, the well-being of the person who is doing the assay. Also, the innards of the sensor should not be damaged on exposure to a particular sample. Alrighty. So now let's look at air and water, how we use sensors for air and water monitoring. The primary measurement media is air, water and air for environmental detection, but the variety of target analytes is vast. This is very, very important. The variety of target analytes is vast. So we're not looking for 5 targets or 10 targets. We have at least 15 different pesticides, equal number of heavy metals. Then we have much larger number of antimicrobial resistant genes, and then we have so much NOM to measure, and then we have so many other organic materials to measure, so many dyes to measure. So the analytes, the domain of analytes is vast. At sites of potential pollution, such as in factory effluent, so if we have a factory, we know it's making 3 kinds of dyes, it's making 4 different kinds of pollutants. So it will work well for us if we have a biosensor that detects the 4 pollutants. We don't need the one that detects 20, 50, 100 different kinds of targets. So it is desirable in sites of potential pollution, such as factory effluent, where we are already expecting pollution, potential pollution. It is desirable to install online, means data goes to internet directly, real-time monitoring and alarm targeted at specific analytes. But in many cases, random or discrete monitoring of both target species or general hazardous compound would be sufficient. The survey of market potential has identified the increasing significance of this area, and this is now substantiated by a strong interest from industry. I want to give you an example. At the time of recording the first part of this lecture series, there was a very big call from DST, which is DST collaboration with partners in US. It's called IUSSTF, Indo-US Science and Technology Forum, and the call was titled Research Initiative for Real-Time River Water and Air Quality Monitoring. So I am highlighting this to give you an idea that not just we scientists, not just we environmental engineers and students, but even our government and the government of other countries such as US, their emphasis now is on how can we monitor data real time. And when we are talking real time, we don't want to establish big laboratories right next to the river. We just want to have a handheld equipment or a stationary equipment, you install it, you get the data, you are fine. Whether it is river or air both. So, this is immense scope of biosensors here, because biosensors have the ability to be very specific, and even though enzymes and antibodies are very sensitive to interference and temperature changes, we have aptamers, again another bioreceptor, which are very specific and can be better contain their structure as environmental changes happen. Now, another thing I want to mention is in the logo of this particular call, it says Internet of Things. Typically, when we think of biosensors, we want to get the data, maybe the data is displayed on the screen, or maybe it is collected in a system which I can put in my computer and get the data. But if I am interested in real time monitoring, it is very online real time monitoring, it is very important that data is shared right as it is generated with public or with whoever the intended viewers are. In that case, we have something called Internet of Things. So, basically you have your biosensor or any kind of sensor, and then it generates data, and the data is, now let us say generates data, the data is picked up by your mobile phone, which transmits to your big computer in your lab, and then this shows up on your website. Now, this is what is called Internet of Things. I am connecting everything with Internet of Things. Let us say if this shows a warning that hey, something is wrong here, then this mobile will get the information, and then the person working with it will be able to take the right action. Alrighty. So, we have talked about biosensor. There is analyte, there is a target. Now, what kind of targets are possible when we talk of biosensors? You can be talking about proteins, you can be talking about toxins, peptides, vitamins, sugar, metal, and so on and so forth. Basically, now the list is endless. Other xenobiotics. We do not have it here, but we can also detect pathogens. We can detect antimicrobial resistance, all by using your biosensors. Now, proteins, toxins, the example of proteins are very simple. Let us say I want to detect presence of prion or particular protein that I am interested in, then I might create a biosensor for this. Toxins, for example, I want to do environmental analyses for algal or rather cyanotoxins. There are some cyanobacteria, some algae that produce toxins, and I want to know whether those toxins are present or not. Now, in order to do that, if I have a handheld, ideally handheld monitoring detection system that will tell me the absence presence of toxin, great. Peptides, peptides could be your genes, for example. Peptides could be other small nucleopeptides on the cell wall that you can use as detection target. Then vitamins, sugar or metal ion, and so on and so forth. Next is sample high handling. How do I deliver the analyte to bioreceptor? So, here I have bioreceptor, let us say it is enzyme based. So, we have enzyme sticking to it. Now, how do I, and this is my analyte, how do I send my analyte here? That is a very important question. So, for example, in pH you just dip the electrode into the water, right. So, this is important. For example, when you are doing your glucose measurement, you prick the finger and the blood drop, you carry your finger to the paper strip. So, this is one of the first question we need to answer, how will the analyte come in contact with bioreceptor? Once you have solved this, then another question comes, what if the analyte is very low in quantity? For example, only 1 to 10, let us say only 1 to 10 cells of cryptosporia are present. So, how do I detect them? Because my bioreceptor is not coming in contact, because this is too less, the concentration is too less. In this case, I can increase the concentration. However, on the other hand, if I have too much analytes, I might dilute the sample and then introduce it to my bioreceptor. The next is filtration and selection. I can select for the contaminant, I can filter the contaminant apart. Like for example, I can filter, because I know cryptosporia is quite big cell, I can filter it on 0.45 micron filter paper, and then I will get concentration of cell, and then I can introduce them to my bioreceptor and get a signal. Now, let us look at some examples of biosensor. Obviously, the most common is pregnancy test. Basically, it detects a protein in urine. So, you have biosensors here that are enzymes that are specific to this particular protein. When they interact with each other, they give out signal, and the signal is an optical signal, because the color changes. So, colorimetric signal, and then you can know whether pregnancy test is passing or failing. Next is glucose monitoring devices. We have at least 2 kinds of devices that are popular in our country right now. One is the glucose strip, just prick your finger, put your blood drop here and you get your glucose data here. Now again, this is your biosensor here. There are bioreceptors here that when they interact with glucose, they undergo they transform, they undergo conformational change, which is then read by this transducer and then output as the data here. Next is continuous glucose monitoring devices. Now these are very helpful for type 1 diabetic patients, and what we do is that there are 3-4 disposable sensors that are inserted into the skin, and then these sensors interact with the glucose levels, and when glucose falls below a particular value or interact with the glucose, and whether glucose falls or increases, they undergo transformational change, which is picked up by your transducer and it gives you data here. Next you have your BioCore biosensor platform. So, this is a typical laboratory based platform, and then obviously we have our old time coal miners biosensor. So, I hope you take this as a joke that for humor, that even because canary bird, this is yellow canary by the way, that even the yellow canary bird is a biological agent, a biological entity, and thus taking a yellow canary down into the mine to test the presence and absence of oxygen is using a biosensor. Alrighty, and this is our one of the leading biosensors in the world. It's also being sold in our country right now. It is infectious disease biosensor from RBS, and it's really amazing. There are 2 different kinds, and one is breathalyzer, so you can just breathe into it. It will collect your bioaerosols, and then because this is a biosensor, they will interact with different bioreceptors and then it will give a signal. So, basically it works this way. It will take sample through. Now, if you want to test for tuberculosis, you cough in from one end, and when you cough it collects the samples, and then you place this in your reader to process, and within 2 minutes, it is very rapid, it will give tuberculosis positive or negative. I encourage you to go through their website. So, you can just look up infectious disease biosensor from RBS and learn more about what they do, how they do. Alrighty. Now, what are typical sensing techniques for biosensors? Typical sensing techniques for biosensors include fluorescence, DNA microarray, SPR surface plasma resonance, impedance spectroscopy, scanning probe microscopy, AFM, STM, quartz crystal microbalance, SIRS, which is surface enhanced Raman spectroscopy, and then we have electrochemical biosensors. So, let us go through them one by one. In fluorescence based biosensors, what we have is, you have bioreceptors here, and in your bioreceptor, when you interact with the analyte, when the analyte interacts with the bioreceptor, this unit now, this transformed unit, it has the property of fluorescence, which means that if you shine the right wavelength light on it, it will give you a very measurable, very easily recognizable signal. And a very good example would be what we talked about in the previous class, where we had cells that had yeast cells that had undergone mutation, and because of mutation what happens is that we trigger DNA repair genes. So, if you have lot of active DNA repair genes, they will interact with a particular protein, a particular gene in jellyfish. So, let us just call it jellyfish gene, and together they have the property to have fluorescence. So, if you shine blue laser, what you will get is green laser, and then you know already there is mutation present, potentially cancer present. And then fluorescence based sensors are also used when we are looking at different kind of assays, different kinds of microscopies. Next we have DNA microarray. We have talked about this already, but this is a wonderful opportunity to revisit DNA microarray. So, this is basically your lab on chip, and you have valves, and in each valve already. So, you have wells on your chip, your DNA microarray chip. Now, in each of these you have a particular nucleotide, polynucleotide. Now, this polynucleotide is specific to some other. So, remember how pairing happens in DNA? You have for A, it will combine with a T. If you have a G, it wants to combine with a C. A will not form bond with C, G will not form bond with T, A will not form bond with G, C will not form bond with T. If it's RNA, instead of T we could have U. So, because A, T, G, C bonding is very specific, we can use this as a very specific sensor. So, if I know the sequence of a particular agent, for example, I know the sequence of tuberculosis mycobacteria, then what I can do is I can find its complementary, or at least for one particular part which is specific only to mycobacterium which causes tuberculosis, I can make a complementary strand, stick it in here, and then if there are tuberculosis mycobacteria genes present, they will make a perfect complement, and when they make a complement there is a change in their conformation, and this can be converted into an electronic or electrical signal, and then it will say yes, there was a mycobacterium tuberculosis. Beauty of DNA microarray is that while this particular well is detecting for tuberculosis, this might be detecting for gene resistance genes. This might be looking for other kind of bacterial infections. So, we can test our samples for literally thousands of analytes in one go. So, that is your DNA microarray. Then we have SPR, surface plasmon resonance. Again, you have analyte, you have a bioreceptor, they interact, and then the surface plasmon resonance changes. And when that happens, you can detect it, convert it into electronic signal. Then we have impedance spectroscopy. So, the way impedance spectroscopy works is that again you have a plate and you have your diodes. I won't make many of them. Now, each of these diodes act like receptors, like bioreceptors, and when analyte sticks with them, the impedance, which is resistance to alternating current, by the way, the impedance changes. So, because these are not diode, these are either cathode or anode, and depending on how your structure is, and then that will change the passing of current. So, you can have current based impedance meters or whatever, impedance meters. So, what happens is that you have your bioreceptor here. When the analyte interacts with this, the impedance of this particular cell changes. And once you know already, when one analyte was present, it changed by x amount of time, when 2 were present, changed by 2x, 3x, and so on and so forth, you can get semi quantitative or quantitative data. So, this is how impedance spectroscopy works. Next we have scanning probe microscopy, AFM, STM. Basically what they do is, AFM for example will tell you the surface information based on the charges. For example, if you have a surface here and you have your bioreceptors here, these are bioreceptors. So, you have your bioreceptors here. So, if the analyte comes and sticks with it, it undergoes a change, and when it undergoes a change, your AFM needle, so it is a cantilever with a pin at the top, AFM needle, when it reads the data, when it reads your surface, it will notice, oh, there is some change here. So, now you get your data. So, this is a SPM, scanning probe microscopy, AFM, STM based biosensors. Scanning tunnel microscopy also can be used. Then you have quartz crystal microbalance, works in similar principle. Surface enhanced Raman spectroscopy is a very interesting case. So, let us look into it. So, surface enhanced Raman microscopy allows us to detect a very low, very small, faint Raman signals, but we can enhance them. Typically, way of enhancing Raman signal is by using gold. So, if you have, for example, if you have a cyanotoxin, and this work was done at Virginia Tech. So, if you are interested, you can look up Dr. Peter Wixland's work. He worked on using SIRS, and he is working on using SIRS for detecting different kinds of environmental toxins. So, you have Dr. Peter Wixland, Peter J. Wixland at Virginia Tech. So, he is using SIRS, quite a leading researcher. And so, if you have a cyanotoxin, and if you have gold nanoparticles or gold particles that interact with the cyanotoxin, when you shine your laser and when it gives away a Raman signal, a very weak Raman signal, your Raman signal would be enhanced, and you will be able to get very low detection values, very high sensitivity and specificity for your cyanotoxin. Then we have electrochemical signals. Alrighty. Dear students, this is all for today. In the next class, we will go ahead and we'll look more about biosensors. We'll try to find out what are the different examples of biosensors, perhaps even go through them, the ones that we talked just now, and more. And we'll also see about what are the applications for biosensors, where there is scope, and perhaps consider 1 or 2 applications. So that's all for today. Thank you.